Dear students, welcome to the online classes of biotechnology. Today I will discuss about recombinant DNA technology. So recombinant means to combine two different DNA. If you combine DNA from suppose one species is human, and if you will combine some part of or some gene of this human DNA with the bacterial DNA, and this hybrid DNA or mixture of two different DNA will called recombinant DNA. So here in next slide you can see what is recombinant DNA technology. Actually, recombinant DNA technology refers to joining together of DNA molecule from two different species that are inserted into a host organism to produce new genetic combinations that are value to science, agriculture field, medicine or industry. So recombinant DNA on the other hand is the general name for a piece of DNA that has been created by the combination of at least two strands. So as I already discussed, you can recombine or combine two DNA from different species, and they are DNA molecule formed by laboratory method of genetic recombination. So you can generate them in the lab condition by the molecular cloning. And if you will discuss about the discovery, so first of all, recombinant DNA in living organism was first achieved in 1973 by Herbert Boyer of University of California and Stanley Cohen of Stanford University. So they have used E. coli restriction enzymes to insert foreign DNA into the plasmid. So here I have shown you the steps of recombinant DNA technology. This is the introductory part. So you need many things for the recombinant DNA technology. So you need first of all a vector or plasmid vector. Here you can see this plasmid which contain the ampicillin resistant gene. Apart from that, you also need the gene of interest. Here I have shown the human DNA. So it may be the any DNA of human like insulin or etc. So first of all you have to amplify this gene of interest from the human genomic DNA. Once you have isolated it by the PCR, you have to digest or you have to cut both the vector, which is plasmid vector, you have to cut it with the restriction endonuclease. Using the same restriction endonuclease, you can also cut the human gene of interest. Here I have shown this black color for the human gene of interest. Then you can ligate this gene of interest with the plasmid vector using T4 DNA ligase. So this human DNA is now ligated with the bacterial plasmid or bacterial vector plasmid. So this is recombinant DNA. Now in next step you have to transform this recombinant plasmid inside the E. coli cell. And here you can see this plasmid is inserted inside this E. coli cell. Now you have to plate them in the LB agar plate which contain the ampicillin. So the cells of E. coli which contain this recombinant plasmid can only grow in the ampicillin plate. And finally, bacterial clones carrying many copies of human gene are here can be multiplied. So here you can see how this human gene, I have given you example of insulin gene. Suppose it is expressed in the bacterial system, and this insulin you can isolate from the bacteria. So this is the beauty of recombinant DNA technology. You can express the human protein in the bacterial system. Now the application of recombinant DNA technology. So as I already discussed, recombinant DNA is widely used in biotechnology, medicine and research like agriculture research, medical research and basic molecular biology lab. You can use recombinant DNA technology. The most common application of rDNA technology or recombinant technology is the basic research, in which the technology is important to most current work in biological and biomedical science. Recombinant DNA is used to identify, map, and sequence the gene. So, as you know, the Human Genome Project is also the part of the recombinant DNA technology. Without recombinant DNA technology, the Human Genome Project or any genome project of organism was not possible. So, further, recombinant protein are widely used as reagents in laboratory experiments and to generate antibody probe for examining protein synthesis within cell and organism. So, you can also generate some recombinant protein antibody probes, and as I already discussed, you can also generate the human protein or any other protein in the bacterial system. Many additional practical applications of recombinant are found in industry, food production, human and veterinary medicines, agriculture, and bioengineering. So here I have given some example. Recombinant DNA technology is used to detect the presence of various infection in the person. So as you know, by rDNA technology, you can prepare some molecular probes. So using these probes, you can detect the any pathogen present in human or any living organism. Apart from that, recombinant DNA technology is widely used these days in the agriculture field also. So, for example, you may heard about this BT cotton. So, this BT cotton is protected by the that insect, that is bollworm. So, third application is in medicine. So, as I already discussed, insulin production by DNA, rDNA technology is the classical example in the medicine field. Further, it is also used in gene therapy. So, by using rDNA technology, you can remove any defective gene from the human body or from the animal's body. And finally, clinical diagnosis, like ELISA is an example where the application of rDNA is quite possible. But apart from that, there are some limitations and disadvantages of recombinant DNA technology also, like destruction of native species in environment. So if you are using the rDNA technology in the field of agriculture, so in this way you can destroy some native species from the earth. Further, these modified plants can theoretically give rise to other modified weeds which are difficult to control, and the cross contamination is quite possible between one species to another species, particularly in the plant species where the pollination takes place. So that is quite possible. It can contaminate the native plants also. Further, recombinant organism contaminating the natural environment. The recombinant organism are population of clones which are exactly the same. So if in a field, if you have grown the hybrid plants, if any of that plant is infected by any virus or bacteria or any pathogen, the whole field can be destroyed because they have the same genetic makeup. Further, the creation of superbug is also possible. Superbug is a can be a bacteria which can be resistant to various antibiotics. So that is called superbug. So if antibiotic resistance, multiple drug resistance, it is called multi MDR. So the MDR can be generated in the bacteria which are present in our environment. So that is also dangerous. That superbug cannot be treated. If that superbug can infect the human body, and no antibody can work on that human body. So that is quite dangerous also. Apart from that, there are some ethical concerns also. If you are using recombinant DNA technology in the human, so you can make the clone or you can exactly make the clone of a particular person. So that is not ethical also. And further, many people worry about the safety and modifying food and medicine using recombinant DNA technology. So if you are using some modified food or hybrid food like BT brinjal, BT potato, so that contain the bacterial endotoxin, so it may be harmful for the human body also. So these are the some demerits of recombinant DNA technology. So I have sum up about basic introduction about rDNA technology today. Hope you enjoyed this lecture. If you have any problem, you can ask me on my Google Classroom. So Next lecture I will discuss about the restriction endonucleases and about the plasmid vectors. So till then, please stay at home, stay safe. Thank you.